Awesome. Thank you, Greg. Um, so yeah, just like Greg said, we're first going to start off by finishing up that LME that we kind of left as homework yesterday. So I'm going to navigate myself back to the tutorial, not in the right spot. So I'm going to go down to computing diversity metrics on the side here. And we can skip past core metrics. We already ran that yesterday. But here's our LME. I'm going to kind of run through this pretty quickly because we should have done it, or we did it as homework, but there is some kind of things I wanted to point out that I think are kind of cool about this. This state column is what allows us to look at this data as longitudinal. So, right, it's using state 2 as a time point that it will be assessing like or changes across time, which is really cool. We're also going to be looking at our 2 different groups, our treatment group or our control group. And then it's also using patient ID, which is the individual ID column, which is what it's calling it here, but I typically hear this called as like a subject ID. So that might be what you guys are familiar with. But yeah, what some sort of identifier that allows us to connect samples from the same individual over time. We're going to look at observed features when we're doing this, and that's what that metric is saying. But also kind of a weird thing that I just want to highlight is that this method is actually visualizing an alpha diversity vector as metadata. And so you can kind of see that we hand in 2 files to our metadata parameter, sample metadata.txt, or TSV, sorry, and our observed features vector. And so basically under the hood, it kind of merges those 2 files, the observed features vector and your metadata together, and that allows it to kind of view it as metadata and kind of allow you to investigate your metadata columns and also this alpha diversity vector. So very cool. So kind of depends. Here we're using technically like 3 metadata columns, but in different ways. So like we're using auto FMT group and patient ID, but I do think that like it's 1 metadata column for parameter that it asks for. But there. It adjusts for the factors that we list here, but I don't think it adjusts for like a bunch of confounding variables. Is that correct, Greg? Okay, yeah. So we will see kind of like the interactions of all of these things that we're looking at. So like alpha diversity as the day relative to FMT continues. We'll see alpha diversity as a result of the treatment groups, and then we'll see those kind of combined in those interactions. And that's actually where the fun part starts for this. But yeah, limited in terms of confounding variables that way. Okay, perfect. Thank you, Greg. So now that I've kind of run down this command, we're going to copy this. And I'm going to bring it over to my Jupyter notebook. I'm going to exit out of this so that we can just kind of start on the same page in case anyone isn't in their terminal yet. Also, I'm going to make that smaller. So I'm going to go down to other and click on terminal here. And that should pop up a terminal for you. Sometimes takes a minute for it to realize that I want to do things. But I'm here. And oh, I'm about to make a fatal mistake. I'm going to do it anyway. Can anyone guess what my mistake is before we see the error? Yeah, exactly. I'm not in the right directory, so Chime's about to tell me that my metadata doesn't exist, which it does exist. So that might lead to some confusion. You're like, I totally have this metadata. It exists. But to them, I told it a file path that doesn't. So to fix this, I'm going to go 2024-workshop, CD 2024-workshop. And then I'm going to ls here just to prove to myself that I have metadata. I'm going to exit this out. And there we can see that I will have my sample metadata. And if I'm looking at this command, I can also see that it points to a diversity core metrics folder, which I can see right here. So it looks like I now am in the right directory. And so we'll run that. Uh-huh. Oh, okay. Totally. I'll just do that. But to really quickly reiterate so that we catch it on Zoom, it does seem like you can add additional metadata columns in as random effects, and then you'll be able to kind of explain those random effects in your model. Yeah, here. Comma separated list without spaces of your metadata columns. And it does seem like you can also provide R style formulas for how to deal with those random effects. All right. So I'm gonna, now that we've looked at that, going to pop open our LME observed features visualization that we just made, and I'm going to download it and open it in View. So view.QIIME2.org. So as we're pulling this up, what did you guys find in this LME as you did it at home? Did you guys notice a pattern? What did you think was cool? Did anyone notice if the diversity between the control and treatment are different? Yeah, the slopes totally were different. Exactly. It is kind of a tricky question because as you walk through this, it kind of tells you a little bit of both things. So I'm going to scroll down to this model. We'll talk about it, and then I'm going to scroll back up to the stats table. But so looking at this, we can see totally you're correct. The slopes look different. Here, I'm going to zoom out a little bit so I can see this whole thing. Is that still readable for you guys? Okay. So we can see our control slope tends to go down over time, and that our treatment slope alpha diversity tends to increase over time. And I'll kind of recenter us back in this, back in our like objective of this study, which is that after stem cell transplant, it's so important to regain health, like high alpha diversity in the gut microbiome, because it leads to better clinical outcomes. And so we're trying to see if we can raise that alpha diversity. And here, looking at these trend lines, it looks like maybe the FMT was successful in increasing that alpha diversity compared to the control group, which didn't receive an FMT. Now I'm going to scroll up a little bit to this table. And so we're looking here. That's kind of what I think of as our like typical p-values for this linear model. And what we see is that when we're looking at our auto FMT as a result of treatment, we don't see a significant difference. It doesn't look like control or treatment really have a significantly different alpha diversity. When we look at it under the lens of day relative to FMT, it also doesn't look like there's a significant change there. But when we look at it, the combination effect of day relative to FMT and treatment group, we do see that there's a significant effect there, which lines up with what we were saying about the slopes. Like it doesn't necessarily look like they have significantly different alpha diversity at some time points. Like this time point, it looks like the spread between treatment and control are really similar. But as we compare treatment and time, we can kind of see that the treatment group raises in their alpha diversity slowly over time, and the control doesn't seem to catch up. Some other things I'll just quickly go over is just the model summary. So we can see how many number of observations we had. We can see how many groups we had. I believe this is kind of akin to our subjects, how many subjects we had over time. And then oh no, sorry. I think this is they're using group a lot here. I do think this is subject number. And then this is the minimum amount of time points that a subject had. And this is the maximum amount of time points that a subject had. And then lastly, this is the mean. This is the mean observed features for this data. And we can see what our dependent variable is and methods. So that's kind of my quick overview of linear models and how we use this to look at alpha diversity. Does anyone have any questions about that? Awesome. Well, I guess onwards then. So I'm going to switch over to slides now. So let me get to where the presentation starts. All right, and they that I can turn this on and talk into it so that I can walk around. So I'm going to try that, but if it turns out really bad, I'm not there. All right. Hello. Oh, yay. Perfect. Okay, I'm a walking presenter, so I would feel confined over there. But okay, so today I'm going to be talking about assessing engraftment extent following an FMT. This is kind of a passion project of mine, and so I'm really excited to show you guys why I think it's so important to assess engraftment when we're looking at fecal microbiota transplants or FMTs. So before we get into it, I kind of want to do a terminology checkpoint. So I think we've heard this term a little bit throughout this, but I'm going to be using the term feature a lot. I'm using it because it's a catch-all for what we're talking about. Here, I'm going to be referring to ASVs or sometimes genuses, like a taxon level resolution. But this is kind of a catch-all that can be used to talk about OTUs or metabolomics or like metabolites or anything. So, but right now, if you hear feature, I'm mostly talking about ASVs, and I'll try to clarify if I'm talking about anything else. My next terminology that I'm going to be using is baseline. In FMTs, we consider the baseline sample to be the sample before FMT intervention, and typically the sample before antibiotic intervention as well. What their recipient microbiome looked like before FMT. This is a really important threshold because we want to make sure it doesn't look like their baseline afterwards. And then lastly, I'm going to use the term donated microbiome. You may have heard the term donor. It's kind of the same thing. I like the term donated microbiome though, because we are donating one specific microbiome. The donor's microbiome changes over time. We've seen that the gut microbiome isn't that stable. And so when we're talking about a donor, that's a person whose microbiome changes and not really the exact microbiome that we're donating. So I believe we got to sequence the donated microbiome, and then we have a full understanding of what that microbiome that we're donating looks like. I'm trying to switch my vocabulary only to donated microbiome, but you might hear me slip up because donor is so common, but that's what I mean when I say that. All right. So this is the point where I'm trying to convince you that engraftment extent is really, really important. So here, when we're looking at FMTs, I think we're kind of asking two questions that are rolled up into one. And I think it's really important that we focus on separating these questions and answering both of them. So I think when we typically think about FMTs, we're thinking, did the treatment achieve the desired effect? Like, did our FMT end up raising the alpha diversity of our individuals who received stem cell transplants? That's kind of our clinical outcome there. But I feel like you can't really know the answer to this question without asking, did the microbiome engraft? And here's kind of my example question, or example of this. You may not see a clinical response, but if you don't know if the microbiome engrafted, you may end up saying that an FM, or you may end up saying like, we didn't get any clear insight from this study. But if we know that the microbiome had high engraftment extent, then you're probably in this box of, we didn't see a clinical response. We had a successful FMT engraftment. And so maybe FMT isn't associated with positive clinical outcomes. And so without knowing the answer to both of these questions, you're kind of left in a haze of not knowing exactly what was the failure of your study. And so knowing engraftment extent allows you to better understand what the result of your clinical outcomes were. So I'm throwing around this term engraftment extent a lot, and I'm going to kind of define what criterion I think are needed to prove engraftment extent and what it means. Engraftment extent boils down to how much the microbiome engrafted, but I think there's a lot of criterion that you need to kind of look at to answer this. So my first one is a mouthful: asymmetric chimeric community coalescence. And that really boils down to the fact that we want both of these microbiomes to coalesce into the recipient, right, after the FMT. And we want it to be asymmetric towards the donor. We want the donor's microbiome to show up a lot more than we want the recipient's baseline microbiome to show up. And so that's why it's asymmetric, but we also want to create a network. There should be a stable microbiome, and so we want to see that create kind of a network of like a community in this gut. So I write this down as we expect the microbiome to shift away from the donor, or sorry, I'm backwards. We expect the recipient's microbiome following FMT to shift towards the donated microbiome, and we expect it to shift away from the recipient's baseline microbiome. Another criterion that we need to assess engraftment extent is donated microbiome indicator species or features. And this is specific microbes from the donor that we want to see engraft in our recipient as proof that we have engraftment. And there should be a large percentage of these donated microbiome features. And I would say that it isn't really decided what a large percentage is. So that's why it's vague there. I think that would be something really cool to look in, like what is the threshold for how many donor features you need? Some people, like Aguala et al., say like 17% donor features is enough in the recipient microbiome following FMT, but other people say that you need significantly more. So I think that that's still kind of hotly up for debate. And the last thing, temporal stability. We're trying to replace a person's microbiome. We are not trying to do a temporary intervention into this. And so we want that recipient microbiome to stay different from their baseline and stay like the donor for a long period of time. However, I would say that a unique microbiome does tend to emerge in the recipient after a while following FMT. And that's probably from environmental factors. Like I said at the beginning, gut microbiomes are known to change. They're a pretty variable environment, so it's not that surprising that the donor microbiome doesn't end up being there like the whole time. But we don't have a threshold really set yet for what it means to, like, how long you have to be stable for before that unique microbiome emerges. I would say though that microbial shifts away from the baseline need to be stable. They're like, we never want the recipient to kind of relapse back to their baseline. So that is a shift that needs to be stable. And I am using this term engraftment extent for a very purposeful reason. When we talk about engraftment, this term is actually coming from stem cell transplants. FMT world kind of took that nomenclature. But with stem cell transplants, it's really easy to say what a successful or unsuccessful stem cell transplant was, because when you start seeing the donor marrow make white blood cells, you're like, awesome, success. But with FMTs, we're transferring hundreds or thousands or even more microbes into a recipient. And what does success mean on a binary? If I engraft 99 of the 100 microbes from my donor, did I succeed or did I fail? It's hard to tell. And then if I engrafted 61 of the 100, did I succeed or did I fail? I don't know the answer to that, but I think that's why it's important that we switch our nomenclature away from this like binary of success or failure to a huge spectrum of engraftment extent that we can kind of quantify and understand. So if I see a lot of community coalescence, I see a lot of my donor features, and I see stability in that, I'm probably going to call that high engraftment extent. And when they have positive clinical outcomes, I'm going to say FMT may be correlated to these positive clinical outcomes. Where if we're on the other side and we just see none of that, then I'll probably call that closer to failure, low engraftment extent. And then there's obviously something in the middle here. And again, that threshold for how high of an engraftment extent you need to have in order to see clinical outcomes is still a little fuzzy there. But I do believe that it's a spectrum of engraftment that we can see with microbiomes. So I'm going to go a little bit into each criterion here. I'm going to explain kind of the tools that are used, and then I'm going to show you some plots that you can generate with Q2FMT to assess them. So asymmetric chimeric community coalescence. This is typically investigated with alpha diversity or beta diversity, or something called source tracking. Source tracking is typically used in like ecology to look at like sinks and sources of like streams. Like initially I looked at like a Grand Canyon study that was looking at different sources of water and trying to locate where they ended up in the Grand Canyon using source tracking. But we're kind of using it to see what percentage of the recipient's microbiome is made up by what. Like how many microbes came from the donor? How many came from the baseline? How many came from both? And maybe there's some novel ones that didn't really seem to come from either of those places. And so that's kind of like, we're tracking the sources that end up making the recipient's microbiome after FMT. I would just say here that alpha diversity is a hard one in terms of community coalescence, because I can have the exact same amount of microbes in my gut as someone else, and we'll have the same alpha diversity, but we don't have any of the same microbes that overlap, right? And so I tend to think of this as a feature that you can give or that you can receive from the donated microbiome. Like after FMT, we see a lot of alpha diversity pop up, and it can be a sign of stability, especially if that alpha diversity maintains long-term, even if the microbiome stops looking like the donor's microbiome. All right, so in order to not spoil anything for our session later on, I'm using a different data set, QIIME et al. They were looking at pancreatic cancer, but really all we need to know is kind of like the time points to kind of settle us into this FMT. And so at the very top here in the blue, we are seeing our baseline samples. Before antibiotics, before anything, that is what these recipients look like. And stepping back a little bit, these are actually human into mice FMTs. And so the recipients are mice, and the donated microbiome is a human gut microbiome. And then at the orange time point, we can see our antibiotic time point, which is why we see that shift away from donor. Oh, and right now we're looking at unweighted unifrac distance to donor. So that would be beta diversity, looking at the differences between two gut microbiomes, right? And so here we're looking at their distance to donor. And after that antibiotic time point, we are more different from our donor than our baseline is. Does that make sense to people? Yeah, it's just because we end up wiping out the microbes that probably were in common between the donor and the recipient. And so we end up with a less common microbiome, or less in common in those two microbiomes. And then, as you can see by my green dotted line, we receive an FMT between time points 2 and time points 3. And at that point, we do shift towards our donor, and we become more similar to the donor. And then the last little bit, we bounce kind of back away from the donor. This would typically kind of be a sign to me of like non-stability, basically. And I would kind of look at that. I think that you have to look at multiple factors here. We could look at our two baseline distance and kind of prove that they don't bounce towards their baseline, and that would kind of be a proof of non-stability. Or we could look at alpha diversity and see how that trends over time. So I think this is more just a talking point, like, oh, maybe there's not stability, but not really proof of that. But additionally, these are mice. So I'm not that surprised that after a while, we see that some of the human gut microbiomes maybe just didn't have, like live there effectively and survive there. And so I'm not that surprised that there isn't that much similarity between these microbiomes. All right. My next one is a method by Aguala et al. and it's called proportional engraftment of donor strains. And it kind of is what it sounds like. They're looking at how many donor strains a sample received from their donor, and then how many of them they actually successfully engrafted. And so same study here. So this is our baseline sample at 0. 1 is antibiotic. 2 and 3 are our post-FMT samples indicated by the dotted line. And so here we can see a couple of things, but I want to go into the interactive link. Feel free to, if you're on the slides, pull this up too and explore. Just so I can kind of show some stuff. But basically, if we scroll over, we can kind of see some details about what's being transferred and how much of it's being transferred, which is cool. But we kind of see, I'm going to point out 2 people as my star children in this study, and then 2 people as maybe my not as star children. But so here, 4 from the top, I can't reach it. That is probably our best engraftment and our most stable engraftment, as we can see. It looks like we have, I think it was like not a huge percentage, like 20 out of 70. Yeah, about 20 out of 70 microbes from the donor engrafted. So not a huge percentage, but we do see that it's kind of stable in its engraftment. It maintained those, which is awesome. And according to Awala et al., I think we're over the 17%. And so it may be perfectly high engraftment. It's a little hard to tell with just any one piece of data. Another student I'd like to point out is this one. He looks like he has the best engraftment of them all. And then it kind of fades. That doesn't necessarily indicate non-engraftment, but it does make me want to investigate him more. Just make sure that maybe he got that high alpha diversity that we're expecting and that he's not returning back to his baseline. Lastly, I'd like to point out these 2 right here. It seems like they never engrafted any donor material. And so if they don't have positive clinical outcomes, I would kind of be not that surprised, I guess, because it looks like they are not going to have positive clinical outcomes. I'd also just like to point out this sample size thing here. My way. Sample size box right here. I have this encoded as to include sample size because proportions are so tricky. You kind of like look at them and you're like, oh, I got 100% engrafted. But like 1 out of 1 is a lot less impressive than 100 out of 100. And so I kind of wanted to make a way to like very obviously visually see that some samples just may have a high proportion, but it may not be that many donor features that we're talking about. And so this allows us to see that, you know, this individual here probably had a lot more microbes to engraft than this individual here. Not that that matters, because this person looked like they kind of didn't engraft any of these donor features. All right. Well, thank you guys for exploring that with me. I'll continue on to donated microbiome indicator features. And so there's kind of 2 broad ways to do this that I've seen. One is that you identify features in your donated microbiome that you think are important to see in your recipient, and you find a way to track them. I typically see like linear models here comparing donor to baseline and maybe seeing what features are enriched in the donor that we want to see transferred over to the baseline. And I'll show you an example of that next, but I just kind of want to quickly touch on tracking previously identified indicator features. Now, this isn't a method that's used a lot, but I think it's so cool and really fun. So when you're able to identify features that your donors have that also have been identified in the literature as being associated with your disease group or your treatment group, I think it's so important to try to like integrate that information into your study and say, okay, this study proved that these microbes were really important for the disease. We see them in our donor. Let's make sure they engraft. And that's a great way to just like make sure that we're including science that's out there into our studies and kind of creating reproducible science that allows us to investigate our engraftment just a little bit better than we would have been able to without that previously identified feature. So I'm going to, really quick, I'm going to show you using ANCOMB-BC, comparing the donated microbiome to the baseline. we have to be really specific about using ANCOM BC because we are violating statistical assumptions if we use repeated measures here. So looking at the donor and baseline, they are not dependent on each other yet. But if I used any other time point, I might have dependency issues. So I'm really specific here to look at donor to baseline. But ANCOM BC2, which hopefully will be in QIIME2 soon, is able to handle repeated measures and should be able to look at this in kind of a more holistic way. But I'll just be highlighting how we can look at ANCOM BC right now. So ANCOM BC is a differential abundance method, which means we're looking at which microbes appear more in one group versus another. So here we're looking at our donor, which is called LTS in this study. And we're going to see what's enriched in our donor and depleted in our donor and kind of be able to track that throughout our study. And so these features up here are enriched in our donor compared to our baseline reference. And these features are depleted in our donor compared to our baseline reference, which means that they're enriched in our baseline reference, right? And so we probably want to see that these features go away at the end of our study, or they don't have to necessarily go away, but maybe they're not as predominant. And we kind of hope that the ones up there that are related to our donor end up making it into our recipients. So that's kind of how we would use ANCOM BC to track these important features. Now, this one isn't really about engraftment, but I have to show it because I love it. So feature peds is what I'm calling this. Looks at a feature, here we're using genus, and looks at how many subjects that feature engrafted into. And so what happens here is we're looking at, say, I'm going to choose one that I can pronounce. Lactobacillus. I love lactobacillus. We're looking at lactobacillus and we're wondering, did that consistently engraft across our subject? And we can see, looking at it, no, it didn't. Seems like it wasn't donated to a lot of our subjects, which makes sense because lactobacillus isn't super commensal gut microbiome. But we can see that, that it didn't engraft very well. Let me choose something that maybe did. Lactospiraceae. It seems like that was in our recipients prior to their FMT transplant. But also a lot of our recipients were able to engraft this following FMT, which I think just, highlights maybe what might be successful microbes to engraft. So in ecology, when we talk about rewilding, we talk about kind of throwing random seeds in a forest and hoping that they grow. And we found that rewilding isn't really an effective strategy because we know that plants are environment specific, right? We want to make sure that we tailor those seeds towards the environment that we want them to survive in. And so now they don't really take a rewilding approach, but a specific, I don't know what they call it now, but a specific seeding approach where they take seeds that they know can survive in that area and then throw them randomly in. And that's kind of what I'm trying to get at with here. FMTs are kind of a rewilding approach. We are just throwing microbes at a gut and hoping that they stick. And we don't really know if they will or why they will. And so I'm hoping that we can kind of start tailoring FMTs, start selecting communities that we know engraft, that we know have positive clinical outcomes for specific treatments, and start kind of, giving a specific FMT as opposed to kind of doing this black box approach where we know we're having positive results and it's really cool, but it's not really tailored. Oh, yeah. How do you know if a species increased? So, so this one doesn't really tell you if a species increased. What I do typically is track it over time. I tend to actually use Q2 longitudinal and use my feature table as metadata and hand it in. One one of these features over here that I've identified as being important to the donor, and I'll track that abundance over time and kind of see if I can see a change throughout time to make sure that our recipients end up with some of these microbes. Say that again. In terms of like how many 16S amplicons I see, we tend to not use like raw sequence numbers as a like way to count, just because 16S amplicon are like PCR'd, and so sometimes that amplification isn't really necessarily representative of us getting like high diversity. It's just representative of kind of the tools we're using. But yeah, so the way that we look at that is alpha diversity, make sure we have diversity in our 16S. We look at beta diversity to make sure that those amplicons, those features that we're looking at, are similar between our donor and our recipient after FMT. And then, yeah, like I said, we use this to track specific features after we have identified what's important to us in our study. Does that answer your question? Yeah. Yeah. Mm-hmm. Yeah. Yeah. Totally. Yeah. I think that's just kind of sometimes just a limitation of like what we can see. Like what we're seeing is only, oh yeah. He was mentioning that like in humans, it must be harder to fully understand what their microbiome is looking like because we can only take stool samples. Well, in mouse, you can kind of investigate that whole GI by sampling as much as you want. And it's a great question. I think it's just kind of a limitation of how we have to study humans a little bit. Like we can only see a representation of what we're looking at. And sampling is the representation of the gut microbiome, and sequencing is a representation of that. And so I do like to think that there's so many levels of extract, like, yeah, just so many levels in between what we're actually seeing. And so that's definitely important. And I always think about that when I'm looking at like donor indicator species. I'm like, we see these in the donors, we're not seeing them in the recipient, but there is a chance they were always in the recipient. And that's hard to overcome. There's no way to overcome it with 16S. I think that some people are hoping that they can overcome it with shotgun by having a little bit more specificity in their strains. But again, it'll be a representation of the gut microbiome. And so, yeah, I don't have a great answer for that. I think that you do have to understand that when it comes to human donations, you're also seeing like effects of what the human is doing every day. We can control that a lot better in the mice. So there's kind of like a toss up a little bit when studying that. But also on a downside, mice don't engraft human microbiomes very well. So like a mouse to mouse FMT tends to do pretty good, but a human to mouse tends to be kind of mediocre. And so there is benefits of mice, but there also is like some negatives because we're not full. Again, it's another representation of a human gut microbiome. So we're not fully studying exactly what we want, you know? I talked to someone who had a kind of interesting idea of like basically barcoding their microbes in the gut microbiome so that they could specifically track that the donor, like it was a donor microbe because those were barcoded as the donor. But again, you could miss that if it just wasn't high enough abundance. So I guess it's just kind of a crossing the fingers. All right. Any more questions before I move on to my last section? Awesome. So I've talked about this a lot, so I'll go through it quickly. We need temporal stability when we're talking about FMTs. It's really important that we see this microbiome shift and we see it shift permanently, because otherwise we're just doing a temporary intervention. And we haven't seen that really helps the gut microbiome long term. But I would like to say there isn't really, as you'll see by my next slide, there isn't really methods for looking at temporal stability. But I would say that it's embedded in every single method that I've showed you in this slide, because if you're looking at FMTs non-longitudinally, you're doing it wrong. We need at least a pre-FMT sample and a post-FMT sample in order to understand whether or not this engrafted at all. And so while there isn't really methods for temporal stability, I think it's just a through line of what I've been talking about. We need longitudinal data in order to prove this engraftment. And we need, honestly, more than the 2 samples I just laid out. Like, it's important to see that temporal stability follow through after FMT intervention. And this is just a slide to show how important it is to use the right tools. I really like this. It's adapted from, I can't remember the journal. Do you remember the journal? Microbe. Okay, yeah. So I adapted this from Microbe, but when we're looking at longitudinal data, it's so easy to look at this clustering, like we saw in our PCOA, and say, okay, that doesn't cluster independently, or doesn't cluster significantly differently. We can look at this kind of distribution that I drew, and we can see that they fully overlap, because that's what the cluster looks like, right? They just fully overlap. But if we think about it as the change over time from our baseline to our after FMT sample, we start to see a pattern emerge, right? All of these points are shifting towards the donor. And when you look at that distribution as change over time, you see that it's not null. And this is all just simulated data, so it's not real, but kind of proving the point of, like, we just need to use the right longitudinal tools to look at this study. And if we don't, we kind of get ourselves in a little bit of a bind. Yeah.
That's exactly right. This is really kind of what we were getting at yesterday, where, like, when you know, there's these like high level tools that we're always starting with, like an ordination. Like, there's probably every microbiome paper you read has an ordination plot in it. But until you start thinking about, like, what are the specific questions that I was asking and how do I address those directly with this data? So for example, like using that LME that we did last night and that we looked at this morning, that's when you start to pull out those answers that are most relevant and most exciting with your data set. And so we see a lot of ordinations that look like that one on the top right, and people get discouraged and they think there's no pattern. But it's really because, like, the ordination is this like initial exploratory tool. And there's a lot more to your microbiome data than like that initial ordination that you're looking at.
Yeah, it also makes it like really obvious how important it is to understand your metadata of your study. I've done analysis for collaborators and not fully understood, you know, the study or the outline, and you start looking at, yeah, that subsection PCOA, or that subsection A PCOA, and you're kind of like, well, there's nothing here. But then once you like dive in and understand the metadata and the study better, you kind of really highlight those exciting parts of it. And so we're about to kind of switch over to looking at Q2FMT. So this is my last little pitch about it. Q2FMT has all of the methods that you need to quantify engraftment extent following FMTs, and it's all located within QIIME2. So it has all of the benefits of performing an analysis in QIIME2, which makes it awesome as a software developer who likes to focus on methods that we can use to make bioinformatics awesome and fun and cool for all of our researchers. But I don't like handling all of the provenance and stuff. So it's great that QIIME2 can package that all for community developers. And with that, here's my acknowledgements. Yeah, thanks to all my lab members who've just helped and made this possible project, as well as all of the collaborators. I work with a ton of clinicians who are doing FMT studies and tell me what I need to know about FMT studies. And so if you have any ideas about FMT studies or have done them before, I would love to chat and pick your brain about tools that you need. And ITCR for funding this project, of course. Awesome, yeah. So that's my little Q2FMT spiel. And with that, I'm going to switch over to our tutorial, but I'm also going to turn off this mic so that I don't have to hold it. Ooh, okay. Am I doing good on time? Okay. Perfect. Okay, so I have a couple of little things. Let's recenter ourselves in the tutorial before I start off. So I have my tutorial pulled up. I have my um, Jupyter Notebook terminal Docker thing pulled up. And I'm gonna close out of some tabs just so that I don't confuse myself. Oh, go back to terminal. Yep, okay, so I'm exiting out of my terminal. I notice that it doesn't really exit out because it's telling me that I'm opening, like I will say terminal 3 here, but it doesn't matter. But so I'm on this launcher page. I scroll down and get to terminal. And then it takes a second for it to realize that I'm here, but it will tell me what my working directory is and then give me that $sign. And that's when I'll know that it's ready to go. Does anyone not have the tutorial and their terminal pulled up and need help with that? Perfect. All right. So navigating through the tutorial, I'm going to, on the left-hand side, I'm going to assessing engraftment extent with Q2FMT. And I will kind of skip through this little prose section. If you want to read it, it's basically the presentation that I just gave. So, but a little bit more condensed. So feel free to read it later on. And I'm kind of going to go through what this is. So we're looking at a command called Q2FMT CC. I call it CC because asymmetric chimeric community coalescence seems like kind of a lot to type. So this is CC. It's our pipeline for looking at community coalescence using alpha diversity and beta diversity. We're going to use beta diversity. Can anyone from this command see what beta diversity metric we're going to use? Yeah, we're going to use Jacquard. That's we're giving it a distance matrix, Jacquard distance matrix. Their beta diversity metrics are set up as distance matrix because it has to be between two samples, right? So each value in a distance matrix is the combination of what features are in common with the other one in its row or column. I kind of want to talk a little bit about why I'm using Jacquard here because I think it's a little bit important. So we can use typically unweighted and weighted unifrac are a pretty common diversity metric that I see used. But with FMTs, phylogenetic distance actually becomes kind of a hindrance. So the unweighted the unifracs use phylogenetic distance to kind of say, okay, these microbiomes are similar because they have microbes that are phylogenetically similar. But with FMTs, I don't really care if they're phylogenetically similar. I want to see that I have the donor's features in my microbiome. There isn't really like a lot of explanation for why I would transfer a phylogenetically similar feature to my recipient, right? We expect the feature that the donor has to be the one that ends up in there. And so I think that Jacquard is the best thing to highlight this. Jacquard is a presence absence diversity metric. So it doesn't care if they have the same abundance. They just have to have the same microbes. So we're kind of just saying, like, do these two microbiomes end up with the same microbes? All right. And then as always, we got to include that metadata file. And then you can see my P distance to donor there. Q2FMT CC allows you to do distance to donor or distance to baseline. And so that's where right now we're going to do distance to donor, but that's where you could find that. We're going to statistically compare to baseline, which is what this P compare means. This is a pipeline, and so we are running not only the prep methods to get all of this into Q2FMT, but we're running the relevant statistics based on the fact that we know, since this is FMT data, that your data is dependent on each other. So we're going to run Wilcoxon SRT behind the scenes. And that's what this P-compare talks about, because it's asking kind of what time point to compare to for Wilcoxon SRT. Our next column is our time point column. I named it time points in the metadata. I would like to say that this Q2FMT kind of expects some standardized time points, which the study doesn't exactly have. Without standardized time points, we're not really able to run statistics on whether or not they're the same at a given time point. And so we kind of created some more standardized time points. But it's important to know that as we get later on in the study, they become a little less standardized. Like time point 6 could be anywhere from like 24 days after the FMT to 150. So just a little something to note as we're looking at this. Our reference column is donor sample ID. We saw this a little bit when we looked at our metadata. This is maybe kind of the trickiest column that you have, but we need the exact sample ID for the donor sample in this column so that we can connect which donor was connected to which recipient. Subject column, that patient ID. We've kind of seen this combo a couple of times before. It was in our LME. That patient ID is our subject and how we're going to track our samples across time. We are using a P filter missing references, which just filters out any samples that don't end up having donors. And then our against group is similar to our P compare. It's just telling us what number is our baseline. For us, it's 0. And then we have our outputs, our stats table, which we're going to get, which is a QZA, so we're not going to peek into it. And then our rain cloud plot, which is a QZV, and we are going to peek into that. Yeah. Which thing I'm using for the data normalization? You mean in terms of like rarefying my feature table? Uh-huh. Uh-huh. Totally. Yeah. 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 So I'll answer your first question and then I might have to circle back on your clarification on your second, but I'm using like the same diversity metrics that we generated yesterday. So I'm using the same normalization tactics that we used yesterday. So I'm rarefying to an even sampling depth of 10,000. And then I'm going to take the diversity metric from our core metrics folder and use that. So this is kind of an example of how core metrics ends up being so useful. Like we looked at those QZVs of emperor plots and we were kind of like, okay, well, that didn't really tell us a lot about our data. But all of the QZAs that are generated with core metrics can be taken and used to analyze a lot of other things like our LME or like Q2FMT CC, which is what we're about to do. And that allows us to explore our data while making sure that all of it is, like normalized and that it's all normalized the same way. Like if I run core metrics once, I will have diversity metrics that all went through the same normalization process, which makes it just a little bit easier to not be juggling like, normalizing every time I want to do a statistical analysis and hoping that I did it the same way each time. Yeah, more or less, yeah. Awesome. Yeah, thank you. Did I answer your questions? I just want to make sure before I move on. Awesome. Thank you for your questions. And I think it brings up a great point. Like, I just want to recircle back and just make sure we're all on the same page of like what's going into this. So we generated a bunch of diversity metrics yesterday when we generated core metrics together. I'm using this. Oh, it highlighted all of them, but the last one here. I'm using, Jacquard distance matrix in order to look at this beta diversity analysis that we're doing. And so if we want to find that in our workshop directory, we can by going over to this little finder, or I could do this in the command line too, but I'll do it in Finder and command line so we can see both. But we can go over to this diversity metrics, and you can see here is my Jacquard distance matrix. And that's what we'll be using today. And also, if I wanted to ls here, I would find that I do have a diversity core metrics in there. Oops, there's feedback or something. And CD, diversity, core metrics. I just tabbed that. That's why it was so fast. I'm not that good of a typer. And then if I ls, I can see all of my files. And if I ls hard star, I can just see my Jacquard ones. And so this is a Jacquard distance matrix, gets turned into a PCOA and then visualized as an emperor plot. So that's kind of why we have all of those files there, but we'll just be looking at the distance matrix. I'm going to clear this out before we start on our thing. Okay, so I just moved myself to computing diversity metrics. So I'm going back to assessing engraftment. And I'm going to this command. So I kind of walked us through this command. Does anyone have any questions before we run it? Awesome. So I'm going to do the typical copy paste thing where I go and copy it, walk over to diversity metrics. Oh, I did something wrong again. Do we know what I did wrong? What is my working directory right now? Core metrics. Yeah, I gave it a relative file. I gave it two relative file paths. And so it failed on both accounts. It tells me about my metadata because it checks first. But my sample metadata is not in my core metrics directory. So I'm not managing to hit that. And I gave it a relative file path from our working directory to diversity core metrics and then to my Jacquard distance matrix. And so it won't be able to find that diversity core metrics because it's already in that folder. This is one of those error messages where like I've seen it so many times that when I hit it, I'm like, oh, I'm in the wrong directory. It like doesn't say that to me at all. But when I see it, I'm like, oh, where am I? I don't know. I apparently don't know because I'm pointing out something that I thought was here and it isn't. All right, so do we know how to fix this? How to get back out one directory? Exactly, cd ... So just jump back one directory. And now if I ls, I should see that sample metadata that it told me didn't exist. And I should see that diversity core metrics folder. So they're not lost forever. We're good to go. So I'm gonna paste again and just make sure everything's right. Sometimes when you're pasting a large command, it ends up being kind of funky. and enter. This shouldn't take too long to run, but I'd like to kind of investigate this rain cloud plot with you guys. It's so cool. Big shout out to Evan Boolean in our lab who designed this rain cloud plot. It is one of my favorite visualizations of all time. All right, so I need to navigate out of this directory. So I just clicked on 2020-workshop because I was in that diversity core metric file. And then I got to go to, so this is hard because I can't see the full one, but I want to download the Jacquard rain cloud plot. If you end up downloading the stats one, you'll get information on provenance, but no pretty pictures, which sometimes is helpful, but I'll focus on the pretty pictures for now. Oh, you are totally right. Beautiful. Thank you, Hannah. So if we download this, do the copy download links work? Copy shareable link? Can I try? Okay. So we have this file from the web thing, and I just want to see if we can get from localhost our visualization. Greg was right that we can't. I just was hopeful. But we know how to do it. We'll just download. Let me just talk through that in case we need that. So right click on this, download. We can't copy those links, unfortunately. And then it should end up in your Finder on your computer actually somewhere. And we'll just drag and drop. Awesome. And I'm gonna zoom in just a little bit so that we can talk about this. So I kind of want to describe like our time and place in this FMT. So I'm going to walk us through what all of the rain clouds mean. So I'm going to try to project. I'm so sorry if you don't hold me pick this up. You want me to use the mic. Hannah gave me the look mic. Remember the mic? So I will use the mic. Okay, so our 0 time point. Does anyone remember what nomenclature I used to describe that? Yeah, we call that our baseline. So blue, our beautiful blue rain cloud, that is our baseline rain cloud. And then 1 and 2 are time points. Well, actually 0 is 2, but we don't have to talk about that. 1 and 2 are time points where they're receiving treatment for their stem cell transplant. They're receiving antibiotics. They received their stem cell transplant, and it's kind of the process in between the infusion and the stem cell engraftment. And I know that it's a little complicated because we are talking about 2 types of engraftment here, but we'll mostly focus on FMT engraftment extent after this. So one thing I do notice is that some of them are really similar to their donor. Do you guys see that? Like these guys over here, they're so similar to their donor. But I do notice that as we're looking at these rain clouds, they're hugely spread across the range of diversity to their donor. And I kind of hypothesize that this is, some people are recovering from the stem cell transplant and getting that high diversity microbiome all by themselves and kind of recovering, but we're not really seeing consistency in that. And we haven't really been able to identify why a person would get their diversity back over another person who just never seems to maintain that diversity. So at time point 3, when we see our, or when we get that FMT, we do see that rain cloud, the distribution kind of tightens, right? Like there's some people that are less similar to their baseline than in the previous time point, but have a pretty consistent range, which is good when we're trying to come up with systems. Like we'd like to see that all the treatment affects someone similarly. And then we can kind of see a natural growth back to their donor in here. And then 5 and 6 is where I was kind of warning you that we're not as consistent with our time points. as maybe we'd like to be. So 6 and 5 can be anywhere from like 25 days after the FMT to over 100. And so I suspect that that bounce back that we see away from their donor isn't really a lack of stability, but is them emerging that unique microbiome that is caused by their environment, what they're eating, how they're exercising, that kind of stuff. All right. Well, now that I've kind of given you guys a little bit of a time and place in this, I'd like to take just a couple of minutes, you guys to investigate it on your own, kind of see if we can come up with any cool conclusions. And then I will kind of show you some of my favorite functionality after that. Yeah, it is. It's totally. So these are rain cloud plots, and I think they're the coolest plots in the whole world. Like even not just these ones, but just the idea of rain cloud plots, because they encode so much data variability for our visualization. So everyone is probably familiar with a box plot. I'm going to turn off some of these. Oh, I accidentally clicked into the meeting. So everyone's probably familiar with the box plot. You've probably seen these a million times. I kind of hate them. And that's a strong opinion, but I just think there's so much data variability that I'm not able to see there. And I'd like to know kind of where all the data points land. And so I think that's why we typically see this, right? We kind of see some people put in like a scatter on their box plot that kind of gives you a better idea of how that data variability plays throughout your groupings, which I think is really cool. But then on top of that, we can add our cloud, which is realistically just a half a violin plot on top of our box plot. And that gives us just a little bit more information on the density of those points inside of our group, which I think benefits us here. And sometimes a little bit more in like very dense data, because here, you can kind of see where the points end up laying, and you can probably visualize that density plot all by yourself. But it does allow you to see kind of where the majority of the points are laying and kind of give you that visual indication of that. Oh, yeah. Yeah. And so I haven't shared this part yet, but this is not a typical part of a rain cloud plot. This is unique to just this visualization that Evan Boolean made, but you can't even really see them here, but we call it lightning, which is the connection lines between a point. And so it's hard to sometimes like show this on, because I want to not hide the line at all, but I want to not hide the line with the tooltip, but okay. And so this is what we call lightning, which allows us to track a subject over time. And this is actually a great choice because it actually goes all the way to 6, which was just a random chance because there's not a lot of points that go all the way to our time point 6. But this allows us to track an individual across time. And so it looks like this individual may not have recovered alpha diversity. I'm making a lot of assumptions here just on this visualization, and I would definitely want to back this up with more findings if I was going to publish this. But like, I'm looking at this individual, and after treatment, they are one of the ones that doesn't seem to recover by themselves, or like spontaneous recovery is what I hear it called. And so they may have been the perfect candidate for why we need to do this. Their distance to donor remains really, really low after their intervention and after their engraftment. But after that FMT, we see it absolutely spike up, right? Which is so cool. That's exactly what we're hoping to see after an FMT. And we actually see it continuously trend towards donor. in our last, or in our 2 time points following that. It seems like they don't have, oh, this is a tricky dot because it ends up in the exact same spot that we were expecting our line to be, but they don't actually have a time point 4. You can kind of tell that by the fact that there's a green dot that overlaps the line, but it's not like made bigger when I scrolled over it. But so they, in their time point 3 and time point 5, trend towards the donor, which is a great sign. And then at time point 6, they trend away from the donor, which again, may be a sign of not stability, may be a sign of them emerging their own unique microbiome. And it's they're, I don't know. There's a couple of subjects I knew, so I wasn't quite sure if I knew the date. Yeah. Yeah, like so we have statistics that kind of tell us a little bit about it. Yeah, you're totally right that like at time point 3 is when we see the biggest like change over time. Now, that isn't like that surprising. That's the moment when they had the biggest intervention, right? So yeah, I would say that time point 3 is probably like our biggest change in time, but it also is our kind of our least informative moment for me as a person looking at FMT engraftment, because I guess I'm not in that moment, I'm not sure what has engrafted and what we're just kind of sampling because we gave them an FMT, right? Like I'm not quite sure what is engrafting and staying around or what we're just capturing because they like had the FMT 2 days prior. Does that make sense? And so, yeah, I'm just, I always love to see that big jump, but it doesn't necessarily mean anything to me until I see some stability in that, which we do see, right? Like we have that big jump and we see some stability in it. That makes me really happy. And I would consider that like sign of high engraftment, but again, mandating factors in engraftment extent. All right, we need to look at the statistics because I can show you pretty pictures all day, but I don't believe them unless we have stats to back it up, right? So just putting this table a little bit in context, we are always comparing to time point 0. We're going to compare to our baseline and make sure that there's like kind of a significant change in distance to donor compared to our first sample. So here at time point 0 compared to time point 1, do we see a significant difference in distance to donor from time point 0 to time point 1? No, we don't. We have a p-value of about 0.5, which is not good. Or it is good because we didn't give them the FMT yet. So it'd be kind of surprising if we saw their microbiome significantly change. And sorry, I just want to reiterate that this is on the whole population. We were just looking at a specific individual throughout time, but when we're talking about these statistics, we're talking about the whole population throughout all of the time points. All right, and then, so when do we end up seeing a significant change? Absolutely, you guys are totally right. Yep, and based on the Q value, we're kind of near significant at time point 3. So we'll take that as you will. But by time point 4, by the time where we've taken a couple of weeks post FMT, maybe hopefully that microbiome has kind of stabilized. Everything we see shouldn't be transient because it's been long enough for the gut microbiome to kind of like wipe any transient microbes out. We do see that significance right here. And yeah, even in our Q value, we see significance, which is awesome. It does seem like compared to baseline, we look more similar to our donor than we did prior to FMT, which is so cool. And then we do notice that in 5 and in 6, we're not looking that different from the distance to our donor compared to baseline isn't significant, which again fits with the trend we were seeing here, where we do end up getting more dissimilar to our donor at time points 5 and 6. Again, not a huge issue. For me, raises a little like stability red flag. Is this okay? Do we need to look at it more? I think we do. So what I would do was look at distance to baseline, alpha diversity, some source tracking around those time points, and kind of see if I can prove that this microbiome isn't like their baseline anymore, but is just a unique microbiome developed after FMT intervention. And with that, we have just a little bit more time, if there's not any thing to bring up before lunch, instructors. Okay, yeah, I'll answer questions. Yeah, the figure. Oh yeah, I would love to show. Thank you for asking that question. So this little 3 dots will give you a lot of options for downloading the figure. Saving it as SVG here. I'll save it as a PNG because I don't know how to open it on this computer as an SVG. So I'm just gonna illustrate that as a PNG for 1 second. And this looks a little funky because it's a transparent background to help you like put it on whatever you need it to be on. But this is what our SVG of this figure would look like. And it makes it hopefully really easy to access for you if you want to download this for your visualizations or your paper or your presentation where you're looking at this. Thank you for the question. Oh yeah. Okay, so this is probably an overwhelming look into code, just to give you guys a little heads up. But this is all done in Vega, which allows us to have amazing, amazing interactive visualizations. And so here is a huge Vega spec that allows this all to be rendered. And it's not running, so let me just run it real quick. It's actually the code running behind the scenes the entire time and creates our visualization for us. And oh yeah. So that's hard. Not possible, actually. Sorry. The way we do this is we like using Python, we outline like a bunch of things that we consider important to us. And then we kind of spec those in. Just because Vega isn't very good at Vega isn't the best at creating visualizations that can be like used for any data. So we're kind of using an amalgamation of Python and Vega specs to get our data in a correct format and kind of spec it into this Vega spec. And that allows us to really kind of use the power of Vega in this interactive figure. And in addition, be able to kind of, use it for any type of data we want and not just make like a singular Vega spec that can only be used once. But I will kind of show that even if you don't know Vega and you can't mess around with this, we've tried to make this as like easily editable so that you can just download the PNG and use it in your paper. And so maybe say distance to donor sample ID of patient ID across time points isn't your ideal name for this figure and you want to name it something different. We actually have these title boxes right here that you can edit it. And maybe you want to call it, this is a long title, guys. Typing wasn't working for me. Okay, so maybe you want to call it like community engraftment extent using Tricard distance. I don't know, anything you want. I could call this like figure 1 if that was helpful to me. And that makes this really hopefully really like usable for everyone and makes it so that there's just one less step for our users as you guys transition from QIIME to creating figures that you're like wanting to publish. And if maybe time points is a bad name because it's definitely a very non-informative name, you could say I don't know, FMT. And you could also change this to Jacquard distance to donor, which I always do because it's important to know what distance you're using. And so I tend to change that just so that I know. Any other questions? Yeah, I have. And I honestly think it's really good. So I tried it with a couple of, all subjects would be represented by the lines and yeah, and all samples by the dots. And then all time points like this by the cloud and the box plot. I think it's very effective because it does allow you to see the population as a whole, and it allows you to see the population as a whole and specifically go through and look at time point or subjects that you may know are problematic or not problematic. Maybe they're successful and that's why you want to look at them. And we're not getting into this right now, but we have just enabled this to be able to facet by groups. So say if you want, like maybe you have 2 treatment groups, like the study we were looking at in my presentation, they had short-term survivors and long-term survivors, and they wanted to make sure the engraftment was the same across both. So you're now able to like kind of facet this so that it includes both groups, which I think is cool and was created via feedback that we got from FMT researchers that said, we need this for this to be usable. So another example of me wanting to hear as much feedback as you guys have, because like what you guys need is what we're trying to create, not like software you guys can't use or isn't helpful. All right. Well, I think we're exactly at time, but it doesn't look like lunch is set up yet. So I'm going to tell you guys about my secret optional exercise that you definitely don't have to do if you don't want, but it's really cool and you definitely should do it. So if we scroll a little bit down on this tutorial, I have an optional exercise. I talked a little bit about how if I was wanting to kind of better understand my stability of my sample, one thing I use is alpha diversity a lot, because if I can prove that maybe they don't look like their donor, but that the alpha diversity maintained after that kind of bounce back away from their donor, that to me is a good sign that, maybe a unique microbiome is emerging, but that it's not going back to their baseline, because typically baseline has low alpha diversity. And so my optional exercise here is to try to adapt the above command to use alpha diversity. The answer is hidden in this box, but don't cheat. Try to do it without looking. And it should generate the same rain cloud plot that you can, not the same, it will have a different metric, but a similar rain cloud plot that you guys can investigate. Yeah.
And so one of the purposes with this exercise is we know like a lot of this is copy pasting commands. And so you're not getting a whole lot of practice with constructing your own commands. And so this is an opportunity to do that. And so basically like everything you need to do this, you've already been exposed to. And so you've created the alpha diversity input value. And you've run a very similar command to this. And so you'll just have to make some adaptations. And so this is a great thing to do, like, over lunch, if you want to just take a few minutes. Remember that -- help parameter that might help you figure out, what you want to provide as input here. And yeah, just try it out, and we can maybe even touch on it like in the afternoon if folks want to go through. But like Chloe said, the answer is here hidden from you right now.
Yeah, and we are going to have again an optional Q&A after this session, or like today's session. And so if you guys are interested in learning more about this one during that Q&A, I'm more than happy to stay and talk about Q2FMT as long as you guys would like to talk about Q2FMT.